I'm delighted to be here today to share with you some of the findings that we have from assembling a cattle genome with Oxford Nanopore reads. And the particular breed of cattle that we are talking about today is a gyr. So gyrs are breeds of cattle that are from India, and they are Hindusine cattle, meaning they have this characteristic hump on their back. And they are very important for breeding because they are known to be resistant to diseases that are found in the tropics. They are very, very important for milk production. And because of this, you find them being crossbred with a number of other species, other breeds in the Bos genus, because they belong to Bos. So this makes them very desirable for interbreeding, intercrossing, etc., so that you have those phenotypic traits that you're interested in. Now, at the moment, there is a reference genome on NCBI, and from the karyotype that we have for cattle, it has an interesting karyotype because cattle has 30 chromosomes. That is, you have 29 autosomes and the sex chromosome. And all the autosomes are acrocentric, all of them. The only non-acrocentric chromosome are the sex chromosomes, which are submetacentric. And the implication of this is the acrocentric chromosomes are very repeat-rich. They are very dense with repetitive DNA, especially in the centromeres and the pericentromeric region. So this has been a major challenge for genome assembly with other technologies because it becomes difficult for you to be able to span the repetitive region. So the reads just collapse on top of one another. But with a very long read that spans beyond the length of that repetitive region, you're able to assemble the whole of that repetitive region, having the expected number of copies of the repeat sequence. Now, over time, this has been a challenge to successfully assembling the chromosomes that we have in cattle. But with long reads from Oxford Nanopore, there is an opportunity to be able to assemble contiguous, single contiguous telomere-to-telomere chromosomes for cattle. So this particular assembly is what we have on NCBI as the current reference that is used for Indicine cattle, and it's actually a cross. It's not a pure Indicine cattle. And this is the highest quality assembly that we have, which is why the community uses it as the reference. Now, we see that the assembly size is quite huge, almost the size of the human genome, 2.7 gigabases. And from here, we see high-quality contig N50, which is at about 26 megabases, which is still quite low. But by the time the whole assembly was polished, you have the scaffold N50 increasing up to over 100 megabases. And the completeness of the genome, as estimated by the Busco scores, give us up to 98% completeness of this assembly. This is not a bad assembly, actually, a very good one at that. But then we have an opportunity, like I said, to have more complete assemblies so that we have all the regions of the genome present in the assemblies, which I will show subsequently. Now, for this particular sample, we had a blood sample from a female, and the assays that were produced, we had ultra-long nanopore reads of about 41x coverage. We had duplex reads also from Oxford Nanopore, about 40x coverage, and then we also had POC, which is a chromosome conformation capture technology, and the POC was to enable us to be able to scaffold and at the same time to separate the assemblies into the two haplotypes because cattle is diploid. So these were base called with Dorado, and then the assembler that we use for this particular assembly is VACO. I'll talk a little bit about VACO shortly. So with VACO, we are able to construct a graph, the assembly graph, using the long reads and also the duplex reads. Then from that, you are able to use the chromosome capture technology to be able to face, as it were, to face the chromosomes into the haplotypes that make up the diploid assembly. And using this, VACO does something beautiful that helps you to see the different haplotypes that you have in your diploid assembly by doing this coloring of haplotype, which is what we see in the last row of that image. So in that particular illustration that they have in their publication, we see a separation of the assembly into a paternal and maternal haplotype, as it were. So in our own case, it's just for us to be able to see that we have haplotype 1 and another haplotype, the second one. So this is the bandage plot from our assembly. So VACO gives you also an assembly graph that helps you to visualize the assembly that you have. And from this bandage plot, which represents the sequence that we have in the assembly, you have the chromosomes labeled, as you can see. And then you also have the haplotypes differentiated by color. So one haplotype is shown there in blue, while the second haplotype is shown in red. And from this assembly, we see a tangle where we have the green circle, and the tangle is representing the rDNA array, which was assembled by VACO. This is one feature that we have difficult to also assemble with conventional technologies, because you have a tandem repeat of this array, and if you don't have a read that spans the repeat, you'll not be able to recover the copies of the rDNA array. And in cattle, the rDNA arrays are found on chromosomes 2, 3, 4, 11, and 25. And if you see there, you'll see that the chromosomes that we have, these particular chromosomes are tangled at that rDNA array. So this is an important feature that you're unable to recover, you're unable to assemble when you use other technologies, but with the nanopore reads, we're able to capture that and have a representation of the copies of the rDNA that we have. So this table gives us a comparison of the current reference, then the assembly that we have with Oxford Nanopore reads alone, and also a mix of other technologies. Now, this may not give you it may not give you a screaming information that these Oxford Nanopore reads are so great, so cool, but then I'll point out some important things to you. Now, take note that the reference assembly that we have is a polished assembly, but the assemblies that we have here is a draft assembly, meaning we just got this straight from the assembler. It has not gone through any kind of manual curation. So the quality that you expect compared to a reference that has gone through series of manual curation won't be that great. At the same time, we still have the representation of the comparison there. So with our VACO assembly, we're able to recover 2 telomere-to-telomere chromosomes. One was a telomere-to-telomere scaffold, meaning you had telomeres at the ends of the chromosomes, but then there is at least one gap that VACO couldn't resolve in the assembly. Then the other one was a single contiguous telomere-to-telomere chromosome. Now, with the assembly that is scaffold, it's easy to be able to go through the assembly graph to resolve the tangle that has created that gap. And these are things that we are doing currently to improve the assemblies that we have. Remember, I said they are draft assemblies. Now, the other thing is, we know that from the reference assembly, we never got any telomere sequence. But from our assemblies, at least, we have a telomere sequence at the ends of 2 chromosomes. And this is an improvement that enables us to be able to capture all the features that we have in this assembly. And in terms of completeness, this is Bosco scores for the different assemblies that were compared. And we would see that the highly polished reference assembly that we had a BOSCO score of 98%. For our own unpolished draft assemblies too, we had completeness that is up to 98.4% thereabout, depending on the haplotype you look at. And this already is is attesting to the completeness of the assembly that we have out of the box from the assembler without any other intervention that we have. And this has been made possible by the fact that we are using Oxford Nanopore long reads for these assemblies. Now, the background is for our cattle assemblies, and as we have in so many other eukaryotes, the genome is highly repetitive. And if you try to estimate the repetitive content you have in these genomes, you have nothing less than 50% repeat. How do you capture all these repetitive elements? It's only when you use long reads that are able to assemble through all the different features that we have in these genomes. And with this, we'll be able to assemble high-quality contiguous cattle genome, the GIA to be specific, and we hope that with this, we keep improving on what we have from the use of long reads nanopore technologies for genome assemblies. So I'd like to acknowledge the wonderful team that have been behind this work, my supervisors, collaborators, they're also from Oxford Nanopore for bringing me here to give you this talk, and all the wonderful collaborators that we have also from Oxford Nanopore, as well as the Bovine Pangenome Project and the Ruminant T2T Consortium. Thank you so much for your time.